Warahmatullahi wabarakatuh. My name is Nur Aina Najwa binti Ishak, S75827. Today, I'm going to present to you how marine invertebrate larvae disperse to three interconnected species: metamorphosis, ballast water transport, and biofueling. I will also highlight how climate change and global shipping intensify this issue, and finally propose a sustainable solution. The main reference for this talk is the study of Plankton et al. 2024 on Hydrodes elegans, a biofueling tube worm. Understanding more about larval dispersal, so we were going to move to Q1, which is understanding marine larval dispersal first. So let's see what we got. Marine invertebrate larvae are the planktonic early stages of benthic animals. Metamorphosis is the transformation from a free-swimming larvae into a silk-settled juvenile. This stage is crucial because it determines survival, dispersal, and colonization of new habitats. Environmental cues strongly influence sentiment. For Hydrodes elegans, bacterial biofilm are essential. This study shows that the O-antigen polysaccharide of bacterial lipopolysaccharide is the key inductive cue for metamorphosis. Without these cues, larvae fail to settle. Examples of species with planktonic larvae include Barnacus balanus, tubworms, Hydrodes, and mussel Mytilus. Ballast water is used in ship maintenance stability. However, it unintentionally transports marine larvae across region. This mechanism is ship pump in ballast water containing larvae, then transport across ocean, and the larvae is discharged at destination and larvae settle and metamorphose. This has introduced invasive species such as zebra mussel and Asian carp into the new ecosystem. This invasion disrupts biodiversity, damages fisheries, and causes billions of economic loss worldwide. Biofueling is the accumulation and requirement of marine organisms, such as invertebrate larvae, onto submerged surface like ship hulls and industrial equipment. The process begins almost immediately when bacterial biofilm rapidly coat any new surfaces submerged in the ocean. This biofilm creates a surface-bound environment that provides incentive cues to small, mobile larvae drifting into the water column. This is Plankton et al. 2034. Larvae like Hydrodes elegans settle on ship hulls and forming dense biofilm mats. Biofueling increases drag on ships, damage aquaculture gear, and alternative ecosystem. Change alters larval dispersal. Ocean warming accelerates larval metabolism but reduces survival. Acidification interferes with metamorphosis success. In the Arctic, warming water allows walrus species to expand northwards, and shown Albert Discotex et al. 2022. This increases the risk of invasive species establishing in new regions. Climate change increasingly rapidly for southern species. While warming temperature can speed up larval development, this may paradoxically reduce dispersal distance by shortening the duration larvae spend in the planktonic stage. Warming conditions can alter the species composition and biochemical fingerprint of bacterial biofilms. Because larvae rely on this species fingerprint to identify suitable habitat, changes in biofilm may influence where and how successfully larvae can metamorphose. By Discotex et al. 2022, warming sea allow border species to expand northwards, such as invasive species. I'm gonna talk about global shipping and invasive species issue. Global shipping intensifies bioinvasions. More ships mean more ballast water discharge and more surface for biofueling. International concerns have led to regulations such as the IMO Ballast Water Management Convention 2017, which required ships to treat ballast water and biofueling guidelines to reduce larval settlement. Current management faces significant hurdles, as evident by the fact that 30 until 45% of larval taxa found in part of Barents Sea are non-local, meaning their adults have yet not established population in those specific areas. This suggests that current mitigation strategy cannot entirely prevent the inflow of southern species. Limitation and the potential improvement. A primary challenge is the difficulty in identifying larvae to the species level without molecular methods, which are necessary to detect non-local species early. Furthermore, there is a lack of data on the true geographical distribution of many taxa, such as Naudi branch, which are often too rare or small for the sampling method like throw or guide. We will move on to the potential future solution for Q3. Innovation solution and technologies. For one powerful tool is DNA metabarcoding. By sequencing the COI genes with high throughput methods, scientists can quickly identify entire larval communities. This allows us to detect invasive species that were previously impossible to distinguish morphologically. Another innovation is particle tracking analysis. Using hydrodynamic models, researchers can predict the likely origin of drifting larvae and identify which borer taxa have the potential to reach the Arctic. This helps us anticipate future invasion before they occur. Next, we will move to environmentally friendly approaches. Research into the molecular nature of settlement cues is also crucial. For example, the O antigen in bacterial lipopolysaccharide (LPS) acts as a signal for larvae like Hydrodes elegans to settle. This stated in Plankton et al. 2024. By understanding these receptor interactions, we can design targeted anti-fouling strategies that disrupt the motif larvae use to identify suitable habitats. This approach is far more environmentally friendly than toxic paint. Lastly, sustainable ocean management. Finally, sustainable management requires collaboration, monitoring outward progress. With this, scientists must continue survey of benthic adults and pelagic larvae to track borer species moving into the Arctic. This data in this context et al. 2022. Protecting benthic health. Government and industry need to understand the role of biofilms in maintaining resilient benthic communities, especially as climate change threatens ecosystem stability. Together, this strategy combines cutting-edge science with responsible management to safeguard marine biodiversity. That's all from me. Thank you for the cooperation.